in the reaction of acetic anhydrides, we have 3 test tubes, which contains the test tube A has a water, the test tube B has an ethanol, and the test tube C has aniline. So for the test tube 1, which has the water, we add 1 ml of acetic anhydride, then place in warm water bath for 3 minutes, and then test it for the litmus paper or the blue litmus paper. So for the observations, the odor has strong vinegar like odor when the acetic anhydride is added in the test tube. And then next is we add the blue litmus paper on it, and as we observed, when the blue litmus paper, the mixture turns into red, which indicates that the mixture is acid. So the product produced, this is a structure of acetic acid. So the next is the test tube B with ethanol. This is immersed in ice water bath, same kaganina. I compared kaganina nga warm water bath. There is a second test tube or test tube B. We immersed it in ice water bath, and then treated with 1 ml of acetic anhydride followed by 1 ml of sodium hydroxide solution. So these are the observations. In terms of the odor, it has different smells noticeable during the addition of acetic anhydride and sodium hydroxide. And then we observe the clear liquid, and it has a white precipitate and black solid substance. So for the product produced, is ester, and this is the structure of ester. So lastly, we have the test tube C with aniline. In this test tube C, we also immerse it in ice water bath and add some 1 ml of acetic anhydride and followed by 1 ml of sodium hydroxide. So in this mixture, we observe that it has a bubbly reaction and also the clear liquid appearance. So this is the structure of the product produced. This structure is what we call acetanilide.
At this moment, I'm going to discuss the expected reaction of a compound when it is mixed with amides. In this, we use the acetamide, which is an amide, functional group containing carbonyl group, or C double bond O. Bonded with a base like sodium hydroxide, acetamide undergoes hydrolysis, breaking the amide bond, releasing ammonia or NH3, which has a characteristic pungent odor. Next, when heated with an acid like dilute sulfuric acid, acetamide also undergoes hydrolysis. The amide bond breaks and the carbonyl group of acetamide react with the acid to form acetic acid, which has a vinegar-like odor.
For the 5th experiment, we conducted preparation of esters. Alcohols were added to different test acids. Acetic acid plus ethanol is ethyl acetate with plastic bubble wintergreen odor. Butyric acid plus butanol is butyl butyrate with pineapple nut cherry odor. Next is benzoic acid plus ethanol, ethyl benzoate with medicinal wintergreen nut cherry odor. Salicylic acid plus methanol is methyl salicylate with medicinal wintergreen odor. And lastly, propionic acid plus propanol is propyl propionate with pear apricot odor.
Now let's move on to the results of experiment 1. For groups 1 to 5, we have positive results for acetic acid's solubility in water, because smaller carboxylic acids, like the acetic acid, have fewer carbon atoms in its chain, so they tend to be more soluble in water. This is because the smaller molecules have a greater surface area to volume ratio, making it easier for water molecules to surround and interact with them. Now for benzoic acid, for groups 1 to 5, we have positive results. However, theoretically, this should be negative. Why? Because benzoic acid has a larger molecular size compared to smaller carboxylic acids like the acetic acid. This larger size can reduce its solubility in water compared to smaller carboxylic acids because of the hydrophobic benzene ring. In benzoic acid, it contributes to its overall hydrophobicity, which can offset the hydrogen bonding interactions with water molecules. However, we may have had gained a false positive result since it is soluble in water to some extent. Now moving on, for butyric acid, for groups 1 to 5 still we have positive results, and theoretically this is correct. The 4-carbon chain in the butyric acid makes it moderately hydrophobic, yet it's soluble in water by facilitation of the presence of the hydrophilic carboxyl group. This group enables favorable interactions that enhance solubility, while larger carboxylic acids like the benzoic ring is hydrophobic. Now, for the citric acid, for groups 1 to 5, we also have positive results, and theoretically this is correct because citric acids exhibit high solubility in water owing to its molecular structure and properties. With 3 carboxyl or the COOH groups and 1 hydroxyl, the OH group, citric acid is notably polar, fostering robust hydrogen bonding with water molecules. So this hydrophilic nature, coupled with its small molecular size, enables efficient dispersion and dissolution in aqueous environments like the water. Additionally, citric acid can undergo partial ionization, yielding H+ ions and citrate ions, which further enhances its solubility through ion-dipole interactions. Lastly, salicylic acid, for groups 1 to 5, we had all negative results, and theoretically this is correct since salicylic acid is sparingly soluble in water due to its molecular structure, which includes a hydrophilic carboxyl OOH group and a hydrophobic benzene ring. While the carboxyl group allows for some interaction with water molecules through hydrogen bonding, the presence of the benzene ring limits its overall solubility. This combination of hydrophilic and hydrophobic components results in moderate solubility, but it's not readily soluble like some other components in water. Moving on, for the alkali solubility, for the acetic acid, it's Na since it's soluble in water, so it did not form salts. So acetic, benzoic, butyric, and citric acid did not form salts in our experiment. However, theoretically, benzoic acid should have formed salts, and we also had a positive result, or we formed salts in salicylic acid. So in benzoic acid, why should it form salts? Because when it interacts with alkalis like sodium hydroxide or the NaOH, it undergoes a neutralization reaction to form sodium salt of benzoic acid, or what we call as sodium benzoate. These salts are more soluble in water than benzoic acid itself. However, benzoic acid does not readily dissolve in alkalis. Rather, it reacts with them to form the soluble salts. Now, for salicylic acid, when it interacts with alkalis like sodium hydroxide, it undergoes neutralization reaction too, just like the benzoic acid. And it is not readily soluble in alkalis. Rather, it forms salts just like the benzoic acid. So for group 1, they had to address or drop 20 drops of NaOH to form salts, while group 2 made 20 drops. Group 3 did not finish this experiment. Group 4 made 25 drops, and group 5 made 23 drops. Now for reversibility. Reversibility means that it could go back to its original states. Now for groups 1 to 5, we have negative results for acetic, benzoic, butyric, and citric acid. However, theoretically, this is sort of Wrong, because the only ones that should be negative are benzoic acid and salicylic acid. Why? Because benzoate and salicylate salts can precipitate in strong acidic conditions, indicating that these have moderate reversibility. While some benzoate and salicylate salts may dissolve back into benzoic acid and salicylic acid and alkali ions under mild acidic conditions, however, strong acids can promote precipitation, and that the insolubility of some salts in strong acids contributes to its low reversibility. The other ones, the acetic, butyric, and citric acid, should have had positive results.
In experiment 2, we measured the pH levels of the various acids. In acetic acid, unfortunately, groups 1, 2, and 3 were unable to perform the experiment. However, in groups 4 and 5, they obtained the pH of 2, which indicates a notable level of acidity. In benzoic acid, only group 4 was able to perform the experiment, obtaining a pH of 3. For butyric acid, groups 1 and 5 recorded a pH of 2, while group 4 recorded a pH of 4. In the case of citric acid, only groups 4 and 5 were able to conduct the experiment, both recording a pH of 1, indicating that it is highly acidic. And unfortunately for salicylic acid, all groups were not able to perform the experiment. Next is the sodium hydroxide titration. In the case of acetic acid, only group 5 was able to conduct the experiment, successfully neutralizing the solution with 13 drops of sodium hydroxide. This suggests that acetic acid possesses a moderate level of acidity. As for benzoic acid, none of the groups was able to perform this experiment. In butyric acid, only group 5 was able to conduct the experiment, successfully neutralizing the solution with 2 drops of sodium hydroxide. This indicates strong acidity. And in citric acid, group 5 neutralized the solution with only 1 drop of sodium hydroxide, indicating that this acid is the most acidic among those tested. And none of the groups was able to perform for salicylic acid. Now let's move on to their acidity rank. Based on pH measurements, both groups 4 and 5 ranked acetic acid as the second most acidic with a pH of 2. As for benzoic acid, only group 4 was able to perform the pH measurement. It was ranked third with a pH of 3. For butyric acid, group 4's pH measurement of 4 positioned butyric acid as the fourth most acidic among the tested acids, while group 5 ranked it as also the second most acidic along with acetic acid with a pH of 2. And lastly, both groups 4 and 5 ranked citric acid as the most acidic among the other acids. Again, no group was able to perform the experiment for salicylic acid.